Greetings, I be to Dr. Muhammad Nur Azlan, the lecturer in charge in supervising me throughout my industrial training. I am Olifaith Anak Holis Miki Langki. Today, I would like to present about my industrial training. I did my internship at TSG Green Sendirian Berhad. The company focuses on planting trees for afforestation projects, as well as to cater for the largest sustainable timber project in Borneo. The company plants, grows, and harvests the trees to reduce the pressure on illegal logging and also logging from natural forest. The company engaged with the local communities, particularly on native lands, to encourage timber tree planting instead of palm oil. The company provides diverse options for local communities on how to utilize their lands. The company provides the tree seedlings for this project. They have production-driven lab and they are located in Kuching, Sarawak. This particular branch was opened in November 2020 and they are focused on the mass micro propagation of timber trees, particularly the species of Paulownia trees. During my internship in TSG Green Sendirian Berhad, I was supervised by Mr. Daryl Nadeng Dominic, who is the lab supervisor. There are 4 specific tasks or activities done in the lab. Firstly is the preparation of culture media for tissue culture. Sugar, myo-inositol, RO water, 5 different stock solutions and gelling agents such as the Galagar are used. After transferring the medium into the 1 liter glass bottle, the agar is added. The bottle cap is loosened prior to autoclaving. The medium is sterilized using the autoclave machine at 121 degrees Celsius for 20 minutes. The molten agar is then poured into the containers in the laminar airflow. 1 liter of media is sufficient for 15 containers. Secondly is the roots transplant. The UV of the laminar airflow is switched on before work starts. The stock explants and media containers are taken from the racks and sprayed with 70% alcohol prior to placing them inside the lamina. All dissecting tools are to be sterilized with the microsterilizer. Roots transplant is done by taking excised roots and culture them in the media in 10 per container. Thirdly, subculturing the explants. The UV of the laminar airflow is switched on before work starts. The stock explants and media containers are taken from the racks and sprayed with 70% alcohol prior to placing them inside the lamina. All dissecting tools are to be sterilized with the microsterilizer, and subculturing the explants is done by removing all the leaves prior to cutting the apical shoots and nodal segments. Apical shoots and nodal segments should be cultured in different containers as they have different growth speed. Fourthly is quality check. The subcultured explants are inspected after 2 weeks periodically. Note for bacterial or fungal contamination, growth performance of plants, and the number of plants per container. Containers with fungal contamination are disregarded immediately. However, explants in the containers with mild bacterial contamination can be saved. This is my schedule of my training program. In week 1, I was asked to prepare sufficient amount of culture media daily. In week 2, I was asked to do the same, which is prepare sufficient amount of culture media daily. In week 3, I was asked to deflush the Paulownia explants and perform roots transplant. In week 4, I did subculturing of the Paulownia explants and deflusking of the Paulownia explants. In week 5, I was asked to do roots transplant and subculturing of the Paulownia explants. In week 6, I performed quality check on the explants. In week 7, I performed quality check on the explants and we all went for tree planting at site. In week 8, I did roots transplant and performed quality check on the plants. And in week 9, I did subculturing of Paulownia explants as well as apply barcode stickers on the racks. My objectives during this internship is to gain knowledge on the usage of general consumables and equipments in the lab. I want to be able to apply the skills and knowledge that I have learned during class at the job. I want to gain experience in working in the real world, and I want to be exposed to professional role models or mentors at the job. Last but not least, I want to achieve better confidence and social skills. These are the photos that I have taken during the preparation of culture media. The picture on the right shows the containers filled with culture media. They are stacked in 5. And the picture on the right is the rack where the containers are arranged. Next, the photo on the left shows the molten agar in the glass bottle prior to pouring them into the containers. And the picture on the right shows my mentor, Sally, who gave me the task to arrange the clean containers into the laminar airflow. Roots transplant. The picture on the left shows me taking the excised roots for roots transplant. And the picture on the right shows me doing the roots transplant in the laminar airflow. These are the apical nodes and roots that I have subcultured. The picture on the left are the 2 laminar airflows that are used meant for pouring the molten agar, and the lamina on the right is for me to do subculturing and also roots transplant. The picture on the left is the autoclave machines, and the picture on the right is the nursery where the mature plants are placed and grown. I was asked to do deflasking at nursery. This is me doing deflasking. Basically, deflasking is the process whereby we take the plants out from the container and we have to remove the agar properly and gently from the roots, and then we are going to plant them in the soil. This is the example. After we deflask, we will plant them in the soil in the pot containing coco peat and peat moss. In these pictures, I was doing quality check. And these are the examples of bacterial contamination. These are the examples of fungal contamination. Okay, so these are the pictures when we were at site doing tree planting. We were asked to plant 100 trees, but on that day we were only able to plant 88 trees. The picture on the right is when the staff teach me how to use the auger drill machine, which is a machine to dig the soil. There are many knowledge and skills that I have obtained during my internship. One of it is I am able to operate general lab equipment such as autoclave machine, the RO water generator, pH meter, laminar cabinet, and the infrared micro sterilizer. I gain knowledge on what roots transplant is about and how it is done. I am able to subculture explants. I'm also able to deflask mature plants. I have deeper understanding on the importance of proper aseptic techniques to avoid or reduce bacterial or fungal contamination during subculturing. I am able to identify contaminated plants and dead plants. I gain more skills in using auger drill machine during tree planting, and I have more knowledge on the differences between bacteria and fungi. There are a few problems that I face. One of it is I measured the wrong amount of consumables during media preparation. Secondly, I spilled the molten agar on the laminar surface. Thirdly, the subcultured explants that I did have a very high contamination rate during the first few weeks. Fourthly, the aseptic techniques are not done properly, which cause contamination in the container. And last but not least, I was not being able to achieve the daily production target, which is 100 containers per day, which is approximately 1,000 explants when doing the subculture. The industrial training experiences that I experienced was the smooth application into the industry. The staffs were friendly and helpful. The supervisor has the willingness to share his knowledge to me, and the supervisor also gives and brief and clear instructions. The company has a very professional and systematic management, and the company also exposes interns to different tasks to do in the lab. In conclusion, I'm able to increase my working experience, specifically working in the lab. I have more confidence in communicating and socializing with new people. I'm able to be more open-minded, and I am able to engage with working environment. Furthermore, I am able to accept and carry out different challenges and tasks that are given to me. This is all from me today. Thank you.